Let's now discuss the digestive process, including absorption, of the three major nutrients that we're ultimately getting from our food through the digestive process. Let's start with polysaccharides, ultimately sugars. When we're first taking food into our mouth, many of the sugars that we take in are in the form of polysaccharide. Ultimately, we want to break these polysaccharides down into smaller and smaller particles so we can then absorb them into our body. Amylases are the major chemical that play a role in breaking down polysaccharides into disaccharides. In this case, we've taken polysaccharides, poly meaning many sugars, and breaking them down into disaccharides, di meaning two sugars, as we can see in the image here. Amylases are released from the salivary glands in the form of salivary amylase, and then the same kind of chemical, amylase, is also released from the pancreas, pancreatic amylase. Sugars are the major chemical that we ultimately get our energy from in our body. And so because they play that major role, we have amylase being released both from the salivary glands and from the pancreas in order to break down as many polysaccharides as we can into disaccharides to get as many sugars out of our food as we possibly can. The disaccharides are then broken down. The chemicals in the small intestine that are released are sucrase, which breaks down the chemical sucrose, lactase, which breaks down the disaccharide lactose, and maltase, which breaks down the disaccharide maltose. Ultimately, once this process has occurred, we've broken down those sugars into monosaccharides, individual sugar molecules. The individual sugar molecules are glucose, galactose, and fructose. The sucrose, lactose, and maltose are made up of various combinations of these monosaccharides. All of this has occurred within the GI tract. But now, how do we absorb these sugars? Sugars ultimately can dissolve in water, and so it's very easy for them to move across our intestinal wall and then move into blood vessels found within the intestinal wall, as we saw earlier on. Taking a look at proteins. Ultimately, a protein is made up of many amino acids, as indicated in the image here. Proteins are broken down by proteases into individual amino acids and then absorbed. Proteases include, in the stomach, pepsin, which breaks down large proteins into smaller pieces of protein. From the pancreas, we release trypsin, which breaks down those smaller portions of protein into even smaller portions. One of the major types of trypsin is chymotrypsin. Chymo referring to chyme, and then trypsin referring to the fact that it breaks down proteins into smaller pieces. The small intestine also releases proteases, which further break down those smaller chunks of protein into individual amino acids. Amino acids are also dissolvable in water, and so they can move across our intestinal wall into blood vessels. Here's a big glob of fat. As we mentioned earlier, fat is not dissolvable within water. And so the first process that we have is the release of bile from the liver that causes emulsification. Emulsification is that process that we mentioned earlier where the fat is actually broken down into smaller bits due to the bile and allows those bits to actually dissolve in water. The liver produces it, as we mentioned earlier. The gallbladder releases it. Then we have lipases. Lipases are released from the pancreas. The lipases actually break down those tiny fat globules, breaking down triglycerides into fatty acids and the glycerol that actually make it up. In this case, the lipase is an enzyme breaking the chemicals down chemically, whereas the bile broke the fat globules into smaller and smaller chunks, however, did not change the chemicals that those globules were made up of in the first place. We mentioned that the proteins, amino acids, and the carbohydrates are absorbed from the intestine into the villi, into the capillaries as shown here. However, as we've mentioned, fat does not dissolve in water, and our blood is made up mostly of water. So fat will cross the intestinal wall into the lacteal, which is the lymphatic portion that we talked about earlier in this unit. The fat goes into the lacteal, then travels along lymphatic capillaries to lymphatic vessels, and ultimately will be returned to the cardiovascular system eventually, but through the right lymphatic duct and the thoracic duct that we discussed during the lymphatic system. It can then travel through the cardiovascular system to be delivered to cells that ultimately need those fat products. We looked at this image earlier, but it's showing the same thing that we just saw in the previous slide. The lacteal that we saw within the villi, and then the blood capillaries as well. So let's discuss the control of digestion. There's two major factors here. The nervous system, specifically the autonomic nervous system and the parasympathetic branch of that, plays a role in the control of digestion. Our senses take in information about the food that we see. Thoughts and memories also play a role. And so if you've ever walked into a room where there's some freshly baked cookies and you have that sweet smell in the air, and you've had delicious cookies before, you think about these things and you're sensing the smell, and maybe you even see the cookies. And what starts to happen is your body starts to get prepared to eat. You start to salivate without even thinking about it. This is the role that the autonomic system plays in controlling digestion and helping to prepare your body to actually digest food that you want to eat, even on a subconscious level. The opposite of this is also the case. If you've ever walked into a room where there's a disgusting smell, the last thing on your mind is probably to eat. You wouldn't start salivating because your body is trying not to prepare yourself for eating, because you wouldn't want to eat something that ultimately might have bacteria or viruses in it and get you sick. Hormones also play a role in controlling digestion. Let's take a look at that now. Here we have a table where we have a number of hormones, the site of production, and the stimulus of production, and then the function that that hormone plays. So we can start with gastrin. Gastro is referring to stomach, and so the site of production is the gastric mucosa, that inner lining of the stomach. The stimulus is the presence of food in the stomach, and also high amounts of protein in the food that's being found in the stomach. The gastric mucosa releases gastrin, and this increases production of gastric juices within the stomach itself. It starts producing the enzymes, proteases, and HCl, and the other chemicals that we've discussed already, in order to help digest the food that has arrived in it. Cholecytokinin. The site of production of this hormone is the duodenal mucosa. So the mucosal layer of the duodenum, that initial portion of the small intestine. The stimulus is high fat content within the chyme that is coming out of the stomach. The prefix of the hormone, chole, is referring to the gallbladder. And so what happens here is the cholecytokinin stimulates the pancreas to produce enzymes that will break down fats. It also, however, stimulates the gallbladder to release the bile to cause the emulsification of fat. And so the cholecytokinin here is referring to that it stimulates the gallbladder to release that bile. And then the second function, it stimulates the pancreas to then release the lipases that ultimately break down the fats. The final hormone is secretin. The site of production is again the duodenal mucosa. Its stimulus is the acidic chyme. As the chyme comes out of the stomach, it's highly acidic because of the hydrochloric acid that was mixed in within the stomach. The primary function of the secretin is to stimulate the pancreas to produce and release sodium bicarbonate. As we mentioned earlier, the bicarbonate acts as a base and will neutralize the acidity of the chyme.